Now we will discuss selection of appropriate strains of probiotic bacteria when they have to be used in the food products or as a pharmaceutical products. While there are a large number of probiotic bacteria and there could be a lot of strains as well, but as a commercial product we cannot use all these different bacteria or different strains as a probiotic culture. So we have to actually look for certain properties, uh, certain functional properties of these microorganisms before they can be actually used as a commercial product. So mostly we would like to do a lot of work before these strains are produced within labs. It needs a lot of research, it needs a lot of efforts and resources to come up with the final product, which is desirable in terms of producing the functional properties or health properties that they confer upon the consumer when these products are used. So mostly we would be looking for functional properties or, or health improving properties of these microorganisms when they have to be used as a probiotic culture or as a probiotic product within the market. If you look at different criteria that has to be used in order for selection of these desirable probiotic cultures, it might be different, such as the general aspect of these microorganisms, like the way they grow, the kind of processing conditions they can withstand. It can also be the origin of these probiotic, because probiotic bacteria, they come from a variety of different sources. So we have to look at the origin from where these strains are isolated. Then we need to look at the identity, like what are these specific strains? Do you need to have any intellectual property on them or not? Similarly, we need to look at these kind of microorganisms which have got potential to resist against mutation. So they do not change their genetic makeup very easily. They would be more desirable. Another thing is the technical aspect, such as the growth aspect or the growth of these microorganisms during processing or handling or different processing conditions. They should be able to withstand different conditions of handling, processing, or distribution, storage. So these factors actually would be playing their role in the viability of these probiotic cultures when they are used by the end user. So talking these five criteria in more detail, if we talk about these general characteristics or general aspect, we have to look that they are resistant against environmental stress. As I said, that when you talk about processing of these things in industry or factory setting, the conditions they may be very harsh for the microorganisms to grow. So we would be looking for the kind of candidates which can resist the harsh environment, harsh treatments, such as higher pH or acidic condition or other temperature conditions as well. As similarly, we have to look at, because these live microorganisms, when they are consumed, the GIT tract or gastrointestinal tract would have again different conditions of pH as well. So they should be able to withstand those GIT conditions in which they have to pass through, and then they have to be viable within our intestine to develop their own microflora within the intestinal system. If you look at the functional aspects or other benefits that we are looking for in these strains are, again, whether they are able to adhere to the mucosa, intestinal mucosa, and once they adhere, the next step, can they make colonies there? Can they actually grow well in that conditions? Can they compete with the other microflora? Because as you know that the intestinal microflora is very much diverse. There are a large number of different microorganisms present. So we want these microbes to actually adhere there, make their colonies, and outnumber the other non-desirable microorganism within our microflora. So we have to look at these characteristics of these probiotics. Then we have to look at whether these strains can outnumber the other pathogens by their own antagonistic potential. Can they actually help to reduce the other pathogenic bacteria or not? Then we again look at the stimulation of immune response, because this is the ultimate benefit that we look for in all the probiotic strains. and the selective simulation of beneficial bacteria, they might help other beneficial bacteria to grow within the intestinal microflora. So these are the qualities or potential that we look for in these microorganisms, which is called as probiotic here. We need to look at the safety of these products as well. For example, they should not be invasive. They should not grow uncontrollably in a living system. They should not transfer any resistance to therapeutic antibiotics. And similarly, they should not have any virulence factor. They should not produce any diseases themselves. If we talk about the other thing, like what are the major obstacles when we have to produce these products commercially, so we have to look at a lot of technical aspects. For example, we need to look at the technical aspects regarding its growth, whether they can grow in that particular condition in which we want them to be subjected. Similarly, are they adaptable to the kind of product that we are finally making out of them? Do they have persistence to stay there in that environment, and the sensitive property of the product, because at the end the consumer is going to like the product. So whether the attributes of that final product, the sensory attributes, are they changed by the addition of the probiotic culture or not? So these are all the things that we need to consider. For example, the most commonly used lactobacillus strains are very much predominantly used in dairy industry for a very long time, almost since 1890 they have been used as a probiotic strains, and they are very much well suited to the GIT tract as well. We know that from our previous experience and all the research experience as well. And if we talk about the selection of these strains from the regulatory point of view, then the regulatory body would be more interested that these products are actually safe. They are not pathogen. They do not create any pathogenicity. They do not create any disease. So these are the two most important factors that has to be taken into consideration by the regulatory authority before they approve any kind of strain for selection as a probiotic product that has to be sold in the market. So this was a little bit introduction about the probiotic strains and what are the requirements to select those strains for a commercial production.